Hi guys, welcome back to Dr. Hill's Academy. Today we'll be discussing glycolysis. Stay tuned. So glycolysis was derived from the two words glykis and lysis. Glykis means what? Sweet. In this context, we are referring to sugar or glucose, while lysis means splitting. So glycolysis is the splitting of glucose. So I'll illustrate it over here. You see that there is glucose, and then we want to form what? ATP. That's your energy. All right. So you're taking food in order to get what? Energy, right? And then there's another intermediate form which is called what? Pyruvate. This pyruvate go to other pathways to further form more energy. But glycolysis produces a particular number of ATP. Okay. And glycolysis, where does it occur? It occurs here in the cell. So this is the cell I illustrated over here. Within the cell, there's the cytoplasm, and then there's the nucleus, which I did not illustrate over here, because glycolysis occurs in the cytoplasm of virtually all cells. So what do we need? We need glucose in this cell, right? In the cytoplasm of this cell for it to carry out what? Glycolysis. And where does the cell get the glucose? It needs glucose to be in the blood, which now moves to the interstitial fluid and into the cytoplasm of the cell. But how does this glucose get to the cell? There are various mechanisms. One, which most of you know about, is what? Exogenous. So we have what? Exogenous routes. Exogenous routes. And then the exogenous route is the process by which we eat food. Over here, you're taking your food, the food comes to the stomach, enters the small intestine, it gets digested, and then is what? Absorbed into the blood. So we have our glucose over here. This is it too. Okay, there are other routes through which we can get what? Glucose in the blood. After you've eaten, some hours after you've eaten, there will be little glucose in the blood. All right, and then your liver senses it and says, oh, I need to get glucose into the blood so that the other cells can utilize it and produce what? Energy. So the liver normally stores glucose as what? Glycogen, right? That's correct. So there is glycogen stored in the liver, and then the liver breaks down this glycogen through a process called what? Glycogenolysis. Okay, so it's broken down to what? Glucose. This is the glucose molecule. Okay, so the liver also takes up other molecules from the blood. For instance, it takes up this. Let's say these are lactates. So it takes up blood lactate and produces what? Glucose. All right. It can also take up something else within the blood, and this is what? Glycerol. Where is glycerol got from? Glycerol is got from what? Fats. So your triglyceride is broken down into what? Glycerol and fatty acids, right? So the glycerol is broken down into what? Glucose by the liver. And then there are other amino acids. There are some amino acids within the blood. So amino acids that can be broken down into what? Glucose. So we've said that there is breakdown of what? Hepatic glycogen. Another route is what? Your lactate. As your lactate is being converted to what? Glucose by the liver. Another one is what? Glycerol being converted to glucose by the liver. And then another one is what? Amino acids. So that's the glucogenic amino acids. So these three are converted to glucose via a process known as what? Gluconeogenesis. And then this one, that's hepatic glycogen, is converted by what? Glycogenolysis. So gluconeogenesis means formation of glucose. That's new formation of glucose, okay, or formation of new glucose molecules from other sources. All right, then glycogenolysis or glycogenolysis, some people would call it, is breaking down of what? Glycogen. So glycogen is a polymer of glucose stored in the liver, and is also stored in muscles, skeletal muscles. Okay, so the liver breaks down its glycogen when it notices that the blood is short of glucose. So it breaks it down so that other cells can utilize the glucose for what? Energy. Then the other one is the exogenous routes, which we talked about. So now we've had enough glucose in the blood. What do we need? We need to bring this glucose to the interstitial fluid, and then the glucose diffuses into this interstitial fluid over here. So we have our glucose in the interstitial fluid, right? Now we need the glucose to enter here. How does the glucose enter there? You know, for a molecule to cross the membrane, that's the cell membrane, it needs to what? Be so small, or it needs to be what? Lipophilic. That it needs to be what? Lipid soluble. Glucose is not any of those. So how does it go into the membrane? It needs to have transporters via which it can go into the cell. So there is a transporter here which allows glucose to enter. This transporter is called what? GLUTs. So GLUT. That means glucose transporters, right? So there are about 14 isoforms of glucose transporter. I'll tell you about the four most important ones you need to know. Okay, so they are the what? GLUT 1, GLUT 2, GLUT 3, and GLUT 4. So I want to tell you a simple mnemonic which you use to know all these glucose transporters. You know, they are labeled or they are named based on where they are found. All right. So GLUT1 is found in particular places, GLUT2 is found in particular places, just like that. Okay. So B, 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 kids' lips are pink independently, but fathers and mothers depend on lip balm. Okay, so we have BBB, kids' lips are pink, but fathers and mothers depend on lip balm. Are kids' lips pink? Well, kids these days also use lip balm, all right? So I don't trust them these days. So we have BBB, kids' lips are pink independently, but fathers and mothers depend on lip balm. BBB for what? GLUT1, kids' lips for GLUT2, pink for GLUT3, and fathers and mothers for what? GLUT4. So BBB kids' lips are pink independently. Why did I say independently? Because they're independent of what's insulin, right? So fathers and mothers depend on lip balm. Please don't mind this lip balm. Is what? Insulin. So you can see that the GLUT4 cells, that's the cells that have GLUT4, depend on what? Insulin. Let's explain that. Imagine insulin molecule comes over here. There would be intracellular signaling mechanism. Okay, that would cause activation of certain proteins. But first of all, before that, there would be vesicles that are packed with what? GLUT4 over here. So I'm drawing GLUT4. So these are your GLUT4 molecules. And then these GLUT4 are packed with what? Vesicles. So these are vesicle associated what? GLUT4 molecules. So the signaling mechanism causes what? Activation of certain proteins. And these proteins cause the fusion of this vesicle with the membrane. When there is fusion of vesicle with the membrane, what do you think will happen? They, they would be together, so they will be fused together, and then there will now be increased expression of the GLUT4 on the cell membrane. When there are more GLUT4 on the cell membrane, there is increased number of transporters through which glucose can enter the cell. Okay, and then there will now be what? Increased influx of what? Glucose into the cell. All right. This will go for the GLUT1, this will go for the GLUT2, this will go for the GLUT3, and this will go for the what? GLUT4. So we have GLUT1, which is the first B, that's what? Baby, that's fetus. So you can see that fetal cells, many fetal cells have what? GLUT1. Then we have the blood, which is your what? RBC. So you can see that the RBC has what? GLUT1. And then the third one is what? BBB. What a coincidence, right? So the second one is You take this K over here, and then you take S and L and P. Okay, this P and L. So we have kidney, we have small intestine, we have liver, and we have what? Pancreas. And then the third one, that's GLUT3, make this capital letter and this capital letter. So we have what? Placenta, N, which is neuron, K, which is what? Kidney. You've seen kidney twice, right? So kidney also has GLUT2, it has GLUT3. Okay, so the GLUT4 is which one? Fathers and mothers, right? So we have our fats and we have our what? Muscles. Okay, so the muscle cells, majorly what? Skeletal muscle and cardiac muscle. So we can see that the GLUT1 is found in the fetal cells, blood cells, that's red blood cell, and BBB. GLUT2, kidney, small intestine, liver, pancreas. GLUT3, placenta, neuron, kidney. GLUT4, fats and muscles. Majorly skeletal muscle and what? Cardiac muscle. Let me tell you one more bonus. So we have what? GLUT5. This GLUT5 is majorly for the transport of what? Fructose. Pretty easy, right? So 5 for fructose. You get it? We've talked about the GLUTs, that's the glucose transporters. Now, if I tell you this is a red blood cell, what should you tell me? GLUT1, right? So this would be a GLUT1 if this is a what? Red blood cell. So we've had our what? Glucose enter through here. I want to represent glucose with something. Remember that glucose has how many carbons? 2, 3, 4, 5, 6. That's correct. 6 carbons. So glucose has 6 carbon atoms. I would represent it over here. So it has 1, 2, 3, 4, 5, 6 carbon atoms. That's our glucose over here. So something happens. You know that this is a cell, and then there are glucose transporters around the cells. These transporters allow the glucose to move in and what? Out. So if glucose comes in through this transporter, it can go out through the same transporter, can go through other glucose transporters as well. So something happens. All the cells, I told you all the cells carry out what? Glycolysis. So they have an enzyme for committing that glucose into glycolysis, or they have an enzyme for trapping the glucose. Once the enzyme does something to the glucose molecule, it is trapped. Okay, because it converts the glucose to something else that cannot pass through this what? Transporters. Okay, so can someone guess the name of this enzyme? I'll tell you. First of all, just know that glucose is converted to 2, 3, 4, 5, 6. Something. This is what? Phosphate. So a phosphate is added to the glucose to make it what? Glucose 6 phosphates. So this is our glucose 6 phosphate that's been formed. Now, before I tell you the name of this enzyme, I want to tell you the kind of enzymes we encounter throughout our glycolysis. Okay, so I want a situation where, as I'm teaching, you're telling me the enzyme without even knowing it. All right, so just listen carefully. So the major enzymes we're encountering are what? One is your kinase. What does a kinase do? A kinase works hand in hand with what? ATP or ADP to either add a phosphate or remove a phosphate. So ATP and ADP, you know, they are energy currencies, right? So they carry energy. ATP particularly carries enough energy. Now, ATP has 3 phosphate molecules. Okay, adenosine triphosphate. So it's adenosine triphosphate. So your kinase over here adds a phosphate or removes the phosphate using ATP to add, using ADP to remove. Okay, so the next one is what? Isomerase. You know what an isomer is, right? So the isomerase changes the structure of the molecule, okay, to something similar, okay, to an isomer of that molecule. So if glucose, for instance, is changed to something else like fructose, you would say it's an isomerase that did this. Okay. Now, can you tell me the name of this enzyme that did this? Kinase, right? So a kinase added phosphate to this molecule. Now, let's talk about the other ones you see. You see what? Mutase. A mutase just affects the structure of one functional group. Let's say a phosphate from glucose 6 to glucose 4, for instance. Now, let me tell you another one. This is a dehydrogenase. Dehydrogenase carries out reduction oxidation reactions. Okay, so it uses NAD majorly, NAD and NADH. So the dehydrogenase uses NAD, NADH to carry out redox reactions. So the enzyme that converted glucose to glucose 6-phosphate is what? Glucokinase. Please don't say glucose kinase, it's glucokinase. So we shorten it and then put kinase, okay? Or hexokinase. So depending, it could be a glucokinase or it could be a hexokinase, depending on the cell. So a glucokinase is specific for what? Glucose. And a hexokinase is for what? Hexoses. So you see that there are hexoses like glucose, fructose, and the rest. They can be acted upon by what? Hexokinase. While glucokinase acts on what? Glucose, right? Glucokinase is found where? In the liver and beta cells of what? Pancreas. So it's found in the beta cells of the pancreas and what? The liver. Now, this glucokinase differs from hexokinase in the sense that glucokinase require so much glucose. So it has low affinity for what? Glucose. Hexokinase has what? High affinity. So hexokinase, higher affinity, glucokinase, ground affinity, right? That's one difference you need to take note of. The second difference is their location. Whereas hexokinase is found in all cells, glucokinase is found particularly in the liver and beta cells. And then the glucokinase having low affinity has what? High Km. Okay, so the Km is what? Michaelis constant, right? Okay, you know Michaelis-Menten equation, right? So this is the Michaelis constant. I would release a video on enzymes and you understand this better. So it has low affinity, high what? Km. While hexokinase has high affinity, low what? Km. The first one you should remember is affinity, okay? So remember ground affinity, high affinity. Hexokinase has how many isoforms? 4 different isoforms. Okay? I want you to understand this enzyme. It has 4 different isoforms. That's A, B, C, and what? D. Or you can say 1, 2, 3, 4. Now, hexokinase 4 is glucokinase. So glucokinase is a hexokinase 4. All right? Before we move on to the next step of direction, there is something that can go wrong in this step of direction, and that's what makes you a good doctor. You need to know what can go wrong in the pathway. It's not just about memorizing the pathway, right? So remember, I told you that glucokinase is found in the liver and beta cells of the pancreas, and in some individuals, the beta cells of the pancreas have what? A mutated form of what? Glucokinase. So glucokinase is not functioning properly. What would you expect? In the beta cells of the pancreas, the glucose would enter and the glucokinase cannot trap it. So it would go back. And then the pancreas, these beta cells, would see it as, oh, we don't have glucose. So there's no need producing what? Insulin. In beta cells of the pancreas, glucose, remember I said glucose is converted to what? ATP in glycolysis, right? So the glucose, as it passes through the glycolytic pathway, produces what? ATP. And the ATP now facilitates the release of what? Insulin. So that's why these patients will have deficiency of insulin and then they have a condition called what? MODY. Okay, that's maturity onset diabetes of the young, type 2. So this is MODY 2. That's type 2. Okay, so they have maturity onset diabetes of the young, type two, whereby they don't produce what? Insulin. You eat, they don't produce insulin. Okay, I know it's a form of diabetes. Glucose 6 phosphate is acted upon by another enzyme to form what? Something that looks like this. So 4, 5, 6. Okay. So if you notice, it has changed. This is still our phosphate, right? But then the structure of this has changed to another what? Isomer. Okay. So we call this what? Fructose 6 phosphate. Fructose 6 phosphate. It has been changed from glucose to fructose, right? And then which enzyme would you think of? Please tell me phosphoglucose isomerase. All right, phosphoglucose. So this is a phosphoglucose, right? Then isomerase changed the form. Now we'll go over to another step. That's reaction. This reaction is reversible. As this reaction 2 is reversible, reaction 1 is irreversible. So I'll use the red for the irreversible reactions. So this is reaction 1, this is reaction 2. Reversible, reversible. For reaction 3, something is added to our fructose over here. 1, 2, 3, 4, 5, 6. Another phosphate is added at the 1 position. You know this is the 6th position, right? So we have position 1, 2, 3, 4, 5, 6. This is 6th position. That's why it was called what? Glucose 6 phosphate. 1, 2, 3, 4, 5, 6. Now, this another one is added to what? 1 position. So what are we going to call this? You say this should be called what? Fructose 1, 6 what? Bisphosphate. Please don't say biphosphate. Don't say biphosphate. Say bisphosphate. Okay. So I hear a number of people say fructose 1,6 biphosphate or fructose 1,6 diphosphate. Please, it's called fructose 1,6 bisphosphate because the two phosphate groups are attached at different positions. If we have something like this, molecule, carbon, 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 and then something like phosphate, phosphate. You can call this a what? Bi, right? So the two phosphates are attached to each other, but in this case they are apart. Okay, so this one is attached to another carbon, this one is attached to another carbon. That's why it's called what? Fructose 1,6 what? Bisphosphate. Is this a reversible reaction or not? Is what? Irreversible. Okay, so just know that reactions 1, 3, and 10 are irreversible. Right? So we'll still go on. The enzyme that converted food to 6 phosphate, you know, this is position 6, right? 1, 2, 3, 4, 5, 6. The enzyme that converted food to 6 phosphate to fructose 1,6 bisphosphate is what? Can you think? It's a kinase, right? So the what? Kinase, that's our phosphofructokinase. This is one, type 1. Okay, there's phosphofructokinase 2 and phosphofructokinase 1. So this is phosphofructokinase what? 1. That's the name of the enzyme. It's the major regulatory step in this reaction. But we'll talk about the regulation later. So let's go over to the next one. The next reaction is the splitting of this fructose 1,6 bisphosphate into two this way. Okay, so we'll have something like this. And then we still have our first part of the reaction. That's 1, 2, 3. 1, 2, 3. Where is this phosphate attached here? Position 1. And this is now what? Position 3. You know, when we split this, it's now we counted 1, 2, 3. 1, 2, 3, right? So we have 1, 2, 3. And then this is at what? Position 1, 1, 2, 3. This molecule would be called dihydroacetone phosphate. Have you heard of acetone before? Acetone is the smallest ketone. So it's a ketone, okay? So the other one is the glyceraldehyde 3 phosphate. We can call it what? 3P for a short. So the glyceraldehyde 3-phosphate and dihydroacetone phosphate are what? Isomers of each other. And the reaction was catalyzed by an enzyme called what? Aldolase. So this is reaction what? 4. So is it reversible or not? It is reversible. So we have our arrow pointing up and down. I want you to notice that one glucose has been given one molecule each, and then suddenly we're having the fructose 1,6-bisphosphate splitting into two, such that one glucose eventually forms two molecules of these three carbon atom molecules, right? So we have two of these three carbon atom molecules, the dihydroacetone phosphate and what? Glyceraldehyde 3-phosphate. Another enzyme can convert this to this. So we can have this molecule, that's the dihydroacetone phosphate, being converted to what? Glyceraldehyde 3-phosphate. Is a reversible reaction. So let me use my black over here. This is reaction 5, rather. Reaction 5. And what are we going to call this enzyme? Phosphotriose isomerase. So that's the enzyme PTI, phosphotriose isomerase, which converted dihydroacetone phosphate to glyceraldehyde 3-phosphate. Since the reversible reaction, the reaction can go either way, but the reaction favors the forward reaction to glyceraldehyde 3-phosphate. So far, we've studied half of the reaction. There are 10 steps in glycolysis. So we've studied the first 5 steps, and then this is the energy investment phase. The other side, the other 10, that's the 6 to 10, is the what? Energy yielding phase or energy generating phase of glycolysis. So since an energy investment phase, you should know that all those places where phosphate was added and ATP was used. So a molecule of ATP gave what? ADP. And then the phosphate was added. So we utilized energy in this reaction. And then another reaction that required kinase was this step 3. So ATP was used and then ADP was formed and a phosphate was attached here. So far, we've invested how many ATP? 2 ATP in the energy investment phase of glycolysis. So guys, we formed our glyceraldehyde 3-phosphates. So this is our phosphate over here. That's the glyceraldehyde 3-phosphate. Now, the next thing that happens is that there is what? Oxidation of this glyceraldehyde 3-phosphate. What did I tell about the redox reaction? That's the oxidation reduction reaction. Dehydrogenase, right? So the dehydrogenase, glyceraldehyde 3-phosphate what? Dehydrogenase catalyzes this reaction. And I told you dehydrogenase uses what? NAD, right? So NAD plus comes here to be converted to what? NADH. And it takes what? A hydride. So a H minus to add to the what? NAD plus to form what? NADH, right? So the H minus is got from where? Glyceraldehyde 3-phosphate. So I'll tell you what this bad boy over here does. That's the glyceraldehyde 3-phosphate dehydrogenase. So imagine I am glyceraldehyde 3-phosphate what? Dehydrogenase. To my left here is what? Glyceraldehyde 3-phosphate. To my right is what? NAD. Okay, so NAD needs something that glyceraldehyde 3-phosphate has, which is what? Hydride. Okay, so it requires hydride to form what? 2.5 ATP in via oxidative phosphorylation, but we'll talk about that later. So the glyceraldehyde 3-phosphate dehydrogenase, I am the one, and then G3P NAD. Okay, so NAD, that's nicotinamide adenine dinucleotide. So over here, nicotinamide adenine dinucleotide. Okay, so that's our NAD, and these are what? Glyceraldehyde 3-phosphate. So NAD requires something, that's a hydride, which glyceraldehyde 3-phosphate has. And glyceraldehyde 3-phosphate, imagine me taking off his arm to give this guy. He has no arm, but he needs his arm, okay? So what I do something very tricky. I take glyceraldehyde 3-phosphate dehydrogenase. I take off his hydride and give what? I give who? the NAD. And then I take something from the cytoplasm, which is what phosphate, and attach to what the place I remove the hydride from. Okay, so I attach a phosphate to him. I remove his what hydride. So we'll have a phosphate going in to be attached to what glyceraldehyde, glyceraldehyde 3 what phosphate. So we have this guy over here. We still have our phosphate, right? But now we have another phosphate attached here. So we need two phosphates because the more the phosphate molecules attached, splitting the phosphate molecule releases what? Energy. So if we split this one, eventually we generate ATP, split this one generates what? ATP. So we need as much phosphates as possible. But now something can occur, something can happen over here. Let me first write the name of this molecule. This is what? 1,3, right? 1,3 bisphosphoglycerate. Remember, it's not what biphospho, is what bisphospho. We talked about it over there. So it's 1,3 bisphosphoglycerate because the phosphate molecules are what? Apart. All right. Nothing can go wrong in this pathway. In some countries like Bangladesh, I'm sure some of you practicing in Bangladesh. We might have some people watching over from Bangladesh. So you see that in those places they have so much arsenate in their rocks and their waters. Okay, so they have a lot of what arsenate, and then they come down with what? Arsenic poisoning. Arsenic poisoning. I'll tell you what happens in this situation. Remember, I told you I was what? Glyceraldehyde 3 phosphate dehydrogenase, right? And then over here we have our who? Glyceraldehyde 3 phosphate, and then we have who? NAD, that's nicotinamide adenine dinucleotide. Okay, so now NAD requires what? Hydride, which this guy has, as a G3P. And then I take the hydride, I give NAD, right? And then I need to replace that hydride I took. Okay, normally I would want to take a phosphate from the cytoplasm and replace it over there. Okay, so we form a 1,3 bisphosphoglycerate and even have energy, right? But in arsenic poisoning, there is much arsenate within the cell. And me now not knowing the difference between a phosphate and an arsenate, I will just stretch my hand and take something that resembles a phosphate, not knowing it's an arsenate, and then I'll put it over there. And then the person will now have what? One arseno phospho. Okay, let me draw the molecule over here. We still have our phosphate over here, right? That's this phosphate. But then the arsenate I put instead of a phosphate comes over here, right? Because we attached it over here. And this will be what? One arseno, what? 3 phospho, what? Glycerate. So we have our 1 arseno 3 phosphoglycerate. What did I tell you about having more phosphate? It means what? More energy, right? There's more phosphate attached to the compound. We tend to liberate more energy. But in this case, we don't have a phosphate. Instead, we have an arsenate, right? And then this arsenate or this arseno bond, this bond over here, spontaneously breaks off to form what? 3 phosphoglycerates. So you see that eventually it will come down to form 3 phosphoglycerates, which we'll see later. This one eventually forms 3 phosphoglycerates after giving off energy, right? An ATP when this one is broken down. But this one breaks down spontaneously to form the same 3 phosphoglycerates. So you can see that no energy is liberated from this spontaneous degeneration, right? To 3 phosphoglycerates, but an energy, 1 ATP, is liberated from the conversion from 1,3 bisphosphoglycerate to what 3 phosphoglycerate. So you can see that eventually the person will have what? Less energy, right? Less ATP. And then you see the person coming down with what? Fatigue, muscle weakness, and the rest. So this is something else that can go wrong in glycolytic pathway. So remember, we were talking about our reversible and irreversible reactions, and this is what? This is reaction what? 6, okay? This is the 6th step. So it's what? Reversible. Okay, I remember I say 1, 3, 10 are irreversible. So this is what? Reversible. The next step of the reaction is conversion of 1, 3 bisphosphoglycerate into something. Now, watch this. We have 1, 2, 3, right? And then we have our phosphate over here. And in this reaction, this bond was broken off. This bond over here was broken to form what? 3 phosphoglycerate. phosphoglycerate, right? That's 3PG. Okay. So we formed our 3-phosphoglycerate. Now, which enzyme would you think of? Remember that an ADP was utilized here to form what? ATP. So we formed energy, right? We formed ATP. The enzyme, I'm sure you would say kinase, right? So it's a phosphoglycerol kinase or phosphoglycerate kinase. So these are what? Phosphoglycerate kinase. So these are phosphoglycerate kinase, and then this was our ADP converting into what? ATP. So there's a liberation of energy in this step. This is what reaction? This is the seventh reaction. Is it reversible or irreversible? Is reversible. Red blood cells have a particular enzyme which helps them to convert 1,3-bisphosphoglycerates to something else. It's not all the molecules that are undergoing glycolysis that are at this stage that can enter this other pathway. So it's a shunt. It's more like a shunt. Some go through here, while some are shunted to another place and then come back here. Okay. So let me explain that. There's an enzyme that does this, and then in this reaction, 1,3-bisphosphoglycerate is converted to what? 1, 2, 3, and then we still have our what? Phosphate, right? Phosphate. But something happens to this other phosphate. This guy over here, that's the enzyme, shifts this phosphate from position 1 to position 2. Okay. Please tell me the name of this enzyme. Is what? Mutase. It's mutase, right? Because I told you that the mutase changes the position of the phosphate or the functional group from maybe 1 to 2, 3 to 4, whatever. But then there's a change from 1 to position what? 2, right? So this will be what? 2. Let me use the green. This will be what? 2, 3 bisphosphoglycerates. 2,3-BPG. All right, so the enzyme is what? A mutase. That's correct. So we have our phosphoglycerate what? Mutase. You can say bisphosphoglycerate mutase. Okay. So phosphoglycerate mutase converting into what? 2,3-bisphosphoglycerate. Why do we need to convert it to 2,3-bisphosphoglycerate? Why can't we just go straight and form our ATP and pyruvate? Now, red blood cells have what? Beta chains, okay? Their hemoglobin have beta chains. And this 2,3-BPG is a molecule that comes to the beta chains, grabs one, grabs the other, and cross-links them, okay? Cross-linking the two chains so that there'll be release of what? Oxygen. So this happens whenever the blood cells move to a region where there is no oxygen, all right? And then it needs to deliver more oxygen. There's little oxygen. It needs to deliver more oxygen. There's a cross-linking, and then there's more delivery. So the oxygen hemoglobin curve shifts to the what? Right, right? So it shifts the curve to the right so that there'll be increased delivery of oxygen. Now with this 2,3-bisphosphoglycerate converted to 3-phosphoglycerate, a guy comes over, and this guy is called what? Phosphatase. A phosphatase is any enzyme that takes off, yanks a phosphate from a molecule, okay, without generating what? Energy, right? So we would have a phosphate being yanked out from here. We have our phosphate over here. So this is yanked out without generating energy. That's not good, right? Okay. Well, it's good for red blood cells because they need it to deliver more oxygen. But other cells don't do that. Okay. So we formed our 3-phosphoglycerate, and then we need to convert this to another guy. These are reaction 7. We need to convert this to another guy. Then this guy over here would have a phosphate at position what? 2. So we convert this to another guy that has phosphate at position 2 instead of position 3, right? So what can we call this guy? As 2-phosphoglycerate, right? This is a phosphoglycerate mutase. So we can see that this is a bisphosphoglycerate mutase. This is a phosphoglycerate mutase. They are similar enzymes. So this 3-phosphoglycerate is converted to what? 2-phosphoglycerates. Okay, 2-phosphoglycerates. Now, is this reaction reversible? Yes. This reaction what? It. So I said I'll use the black for the reversible reactions, right? So I'll use the black here. This is reaction 8. So these are another reversible reaction. Now the other reaction, we have just 2 more to go, so just be patient, okay? So the other reaction is conversion, that's the dehydration of these 2-phosphoglycerates, okay? So the 2-phosphoglycerate will be dehydrated. A water molecule is removed from 2-phosphoglycerates. So we want to remove water. What's the name of this enzyme that does this? Enolase. Have you heard of the name? Enolase. Enolase is the enzyme that removes a water molecule from 2-phosphoglycerate to form something called what? Phosphoenolpyruvate. Okay, so phosphoenolpyruvate. I'm sure you're saying we are coming to the end. I can see pyruvate. Yes, very soon we'll form our pyruvate. Okay, so the enolase converts 2-phosphoglycerate to phosphoenolpyruvate. You can just call it your PEP. That's the short PEP. This is a reversible reaction, right? So this is reaction 9. There's something important about this enzyme. So if someone wants to do a blood sugar test and then you put the blood in tubes, what can occur? You know, there are still red blood cells inside the blood. While you're transporting it to the lab, the red blood cells are still utilizing what? Glucose. Still utilizing the glucose within the plasma, and they're using it to form ATP, right? Using it to form ATP. However, we use something called fluoride tubes. So we know our fluoride from chemistry, fluoride. This fluoride inhibits this enzyme. And then when we use fluoride tubes, that's tubes that contain fluoride, the fluoride inhibits this enzyme and then preventing glycolysis from continuing. Okay, so the glycolytic pathway is seized, and then the red blood cells stop utilizing the glucose so that you can adequately and effectively measure the blood sugar level. All right. So the final reaction, that's reaction 10, is it reversible or irreversible? It's irreversible. I told you 1, 3, 10 are what? Irreversible. So this will form our what? Pyruvate. And we generate ATP. So ADP is used to generate what? ATP. Sorry, let me use the red for irreversible reaction, right? So ADP will be used to generate what? ATP, and then the enzyme is what? It's a kinase. Remember, we have our phosphate over here. We see our phosphate over here at position 2, right? So it's a phosphoenolpyruvate, and then we will form a pyruvate by yanking off a phosphate from phosphoenolpyruvate. So this will be a what? Kinase. Pyruvate kinase. There's something that could go wrong in this step. This particular reaction 10. In some individuals, they have deficiency of what? Pyruvate kinase. So they don't have enough pyruvate kinase. There's deficiency of the enzyme in their red blood cells. And then they produce what? Less ATP. Okay. So since they produce less ATP, ATP is required for stabilizing the membrane. This membrane stability, ATP is necessary for that. And then it's that stability that ATP gives it its shape, that's the biconcave shape that allows it to also squeeze through capillaries. Okay, so when the enzyme is deficient, there is less energy, there is no maintenance of the membrane, there is lower flexibility, so it cannot pass through capillaries easily. So as it passes through the endothelial slits of the spleen, it's what's grabbed by what? Macrophages. That's the splenic macrophages, which now phagocytose it and then causes what? Hemolysis. So you see what we call what? Hemolytic anemia. That's a kind of anemia in which there's hemolysis, there's destruction of the what? Red blood cells. So far, we've discussed the glycolytic pathway, and then we've seen that a glucose molecule produced what? Dihydroacetone phosphate and what? Glyceraldehyde 3-phosphate. So dihydroacetone phosphate was converted to what? Glyceraldehyde 3-phosphate. And then you see that eventually there's two glyceraldehyde, three phosphate molecules from what? One glucose molecule. So everything that occurred in this reaction, in this side of the board, everything that was illustrated in this side happens twice. Okay. So for this is for one glyceraldehyde, three phosphate. Okay. But it happens twice in reality for one glucose molecule. So this in this step of the reaction, in this step of the reaction, we can see that NAD was used, NADH was produced, right? This NADH that was produced cannot be used again to, if the reaction is continuing, to continue the glycolytic pathway, right? Because the NADH that's been formed has to be converted to NAD for the reaction to continue. That's to happen again for the NAD to be reused. So if this cell has a mitochondria, for instance, let's say this is our mitochondria for the cell. Now, the NADH would go to the mitochondria for to undergo what? Oxidative phosphorylation to form what? 2.5 ATP, and then the NAD will come out. We would have something like this. Let me mark it out over here. NADH converted to what? NAD plus in the presence of what? Oxygen. Okay? So the mitochondria utilizes oxygen to convert NADH to NAD, and then it will form what? 2.5 ATP. Okay? We need this reaction to occur for NAD to be reused for glycolytic processes. So if this reaction does not occur, let's say in cells that lack mitochondria, okay, or cells that have poor perfusion, this reaction does not occur. And then the NADH is now so much. Okay. So the NADH slash NAD ratio is what's increased because there's more what? NADH, right? So since the NADH is so much and it cannot it cannot continue this process, something happens. When the NADH is so much and NAD is so little, the NADH to NAD ratio is what? High, right? And then something happens. The pyruvate is converted to what? Lactate. And NADH is utilized to produce what? NAD plus, which can now continue the reaction. So this NAD that is formed can now ensure that glycolysis continues. Okay. The enzyme involved here is lactate dehydrogenase. So this is a step 11 for anaerobic glycolysis. We call this anaerobic glycolysis. That's glycolysis that occurs in the absence of what? Oxygen, okay? Or glycolysis that occurs in cells without what? Mitochondria, right? So when there's little oxygen, when there's little or no oxygen, anaerobic glycolysis occurs. But aerobic glycolysis stops at what? Reaction 10, that's pyruvate. Some conditions in which anaerobic glycolysis occurs normally. So we have anaerobic glycolysis occurring in some conditions. We have physiological conditions and we have pathological conditions, right? So physiological states, that's in the normal functioning state, and then some disease states. So for physiological conditions, there are some cells that undergo anaerobic glycolysis normally. So that's their default. Cells that have what? Low perfusion, and cells that have what? No mitochondria. So I'll tell you the cells, and then you can use the mnemonic what? Alerts to remember them. All right. So the alert, the A is for what? Anaerobic glycolysis. So the A is for anaerobic glycolysis. The L is for what? Leukocytes. The E is for what? I. And under the I, we have lens and what? Cornea. I'm sure you wouldn't expect blood vessels in the lens and the cornea because they are transparent. You need it for what? Light rays to enter the retina. And then the R for what? Renal medulla and what? RBC. That's the red blood cells. And then the T for what? Testis. So all these tissues have cells that undergo anaerobic glycolysis normally, okay, as their default mode of breaking down glucose. Then another example is in what? Exercising muscles. So when you're jogging, when you're running, doing some aerobic exercises, you notice that your muscles are fatigued. You feel your muscles are fatigued, and that's because lactate is being produced. You know, the muscle cells get less oxygen than they need, so they are carrying out so much work, and then they need so much more oxygen. But then the oxygen is reduced because you can't even take in enough breath. That's why you find yourself gasping. All right, so you can't take in enough oxygen, but your muscles are doing so much work now, producing so much ATP. So the muscles have a what deficit of what oxygen, and then it now undergoes anaerobic glycolysis to form lactate. That's because we need the cells need what NAD, but there's no NAD, so they had to convert NADH to NAD by themselves without the mitochondria, right? Now, pathologically, this can occur in cardiovascular collapse or compromise. Let's say cardiovascular compromise. So when the cardiovascular system is compromised, let's say in cases like coronary artery disease, when we have plaques in the coronary artery, for instance, this is the coronary artery. Let's say this is the coronary artery, and then it's going to the heart. Let's say this is the heart. So if there's a thrombus here, you see that blood is not moving to the heart, and then oxygen is not getting there, right? So there's something that happens, maybe some myocardial ischemia. This occurs, myocardial ischemia occurs, and then eventually forms myocardial infarction, okay? But before then, you start noticing that the cardiac cells will start producing so much what? Lactate, because they're not receiving enough oxygen, and then they have to, produce the NAD by themselves. Right? They have to convert NADH to NAD, and then you see that the lactate will spill into the blood. That's why these patients, when they come to the clinic, you do a metabolic panel, and then you see that there's so much lactate in their blood. So this could be one of the causes. Another condition this can happen is what? Cancer cells. So cancer cells are proliferating so much. They're proliferating so much, and then they require more oxygen than they get. So they have to undergo anaerobic glycolysis to produce what? Lactate. And this effect in cancer cells is called what? Warburg effect. So this is called Warburg effect in cancer cells. Okay, guys, I'm sure you enjoyed this lecture. It's been a long, long ride. I'm sure you pretty much enjoyed it. And thank you for staying to the end. Don't forget to subscribe, hit the like button, drop your comments below, and share to your friends so they'll benefit from it. Thank you, and see you soon.